Okay. Very good evening, everyone. The people who did not watch the yesterday class, the people who did not attended the yesterday class, kindly watch the recorded of the yesterday class without fail. Okay. All right. Everyone joined, right? That's great. That's great. That's great. Okay. That's perfect. So in the yesterday class, if every student quickly recall, friends, we have started discussing about the C3 pathway as well as the C4 pathway. I strongly believe, I hope every one of you remembered it also. Every student very quickly answer my question. Don't get confused. Think properly and answer my question. My question is, in order to produce two glucose molecules, in a C4 plant, how many number of ATP and how many number of NADPH are required? I want every student has to answer the question. In order to produce two glucose molecule in a C4 pathway, how many number of ATP and how many number of NADPH are required? Wonderful, wonderful. So we require 60 ATP and 24 NADPH are required. Right, wonderful, wonderful. Okay. Anyone else is gonna join? All right, all right. Okay. Okay. Now, we will see, Telangana set a question paper, this was happened a few days back. Most of the Telangana students are feeling that this year life sciences question paper is extremely difficult. Okay. But let's see how many of you can answer the questions even without reading. I feel extremely happy if you can answer all these questions, I believe you can answer very easily. Now, from the topic of photosynthesis, they have given 3 questions in the set paper. From the topic of a nitrogen metabolism, they have given 1 question in the question paper. Let's see how many of you can answer this. I believe you answer every question. Okay. Now, let's see the first question. All of you has to answer. Don't be hurry. Okay. Photosynthesis is essentially dash reaction. Quickly. All of you, option 1, option 2, option 3, option 4. Every one of you all people commonly know that the photosynthesis is basically what kind of a reaction, friends? Absolutely, it is a reductive reaction. Is it or not? Very good. Okay, now let's look into the next question. Every single question, whatever we discussed, it came, but I don't know. It's depending on your analysis. Anyway, I'll show you. And this is the second question that they have asked from the photosynthesis. I believe you all remember in the yesterday only we discussed this. Look into this. This is the question. Question number 87. Okay. This is the option 1, option 2, option 3, option 4. I believe every one of you give me the right answer. Okay. The question says that, friends, the manganese stabilizing protein in photosystem 2 is associated with the following. Slowly analyze. Every student. Every one of you. I want everyone to give the answer, not 1 or 2 people. The manganese stabilizing protein in photosystem 2 is associated with the following. Of course, in the yesterday class only we have seen, right? We all people discussed it very clearly also. So I have shown you a diagram. I hope you all people remember. You seen this diagram? How many proteins it contains? D1, D2, C43, C47. This is all present in the P680. Wonderful. So that is the second question they have asked. Now, this is the third question they have asked in the exam. Okay. Now, match the following. Okay. First of all the students tell me, this question is from which concept of a photosynthesis? I don't want answer. First tell me, this question is from what topic of the photosynthesis? Everyone. Wonderful. This is from the C2 cycle. In the yesterday class, whatever the steps that we have discussed, the same steps they have asked in the exam. Tell me all the students. Same steps. There's no change also. Come on, quickly. Slowly, every student slowly analyze the question. Don't be hurry, Saida. Wonderful. So we all know very well, right? The 2-phosphoglycolate converted into glycolate. Correct, na friends? And what happens to this glyoxylate converted into glycine? What happened to the glycerate converted into 3-phosphoglycerate? What happened to the hydroxypyruvate? Finally converted into the glycerate. The same thing which was there in the slide. That's all. There were 3 questions asked from the concept of photosynthesis. All the 3 questions are answerable very easily. I have seen every single question out of 100 questions in the core paper. I have seen it every question. There are total 89 questions directly from the PPT itself. 89 questions. The remaining 11 questions, you have to use your brain, because in one question they have given 8 options, surprisingly. But that kind of questions also you people can answer it. Pratisruti, just refresh, dear. It's the problem from your side. Refresh it. All right. Okay. Now, all the students very quickly answer my question. So don't disturb me for 30 minutes. I'm going to complete the entire photosynthesis right now. Every one of you quickly answer. What is the name of the carbon acceptor in the C4 pathway? Shafia, I'll show you 1 minute. Let me complete. I'll show you. What is the, very good. What is that? PEP, phosphoenol pyruvate. Every student very quickly answer. What is the carbon acceptor in case of a C3 pathway? Every one of you. What is the carbon acceptor in the C3 pathway? The people who joined right now, please watch the recorded. Do not miss it. So the carbon acceptor is what, friends? Undoubtedly RUBP. There's no need to get confused with that particular point. All right. Okay. I believe all the students remember it. So, what is the ultimate conclusion? The ultimate conclusion is that, friends, by using the Kranz anatomy, the C4 plants are successfully skipping the C2 cycle, or successfully skipping the photorespiration process. Now, let's try to understand, friends, how exactly the CAM plants are going to skip or avoid the photorespiration and going to fix the carbon into glucose. Actually, friends, CAM plants having 2 goals. The first goal of the CAM plant is, of course, they have to fix the carbon dioxide into glucose, which means that they have to avoid the photorespiration. And what is the another important and significant function? The very important function is what? Minimize the transpiration. Correct, na? Minimize the transpiration, because every student commonly know that the plants which are actually living in the desert, they have to, I mean to say, they have to prevent the transpiration in order to survive for the long period of time. Is it or not? Of course, yes. Now let's see how CAM plant is going to fix the carbon, how the CAM plants are going to minimize the transpiration, and what are the various examples of the CAM plants? The plants which are actually living or which actually belongs to the category of a Crassulaceae family. Those we generally call it as the CAM plants. Best example, you can remember multiple example: cactus, pineapple, agave, succulents, bromeliads. There are multiple examples are there. Now, again, every student use your brain and keep on answering my questions very correctly, not to get confused. Now, anyway, our ultimate goal is to fix the carbon only, but you might observe many times, if you look into the temperature in the deserts, friends, during the daytime, the temperature is extremely higher. You can't imagine the temperature. It is always above 50 degrees centigrade. But friends, when you look into the night time, all of you know very well that temperature is dramatically fall down. There's no doubt about it. Correct, na? Daytime the temperature is very high and the nighttime temperature is dramatically fall down. So you only think now. So since the temperature is extremely higher during the daytime, if the CAM plant open the stomata during the daytime, what happen? Entire water loss will occur. So that is the reason what the CAM plants will do. They never ever open the stomata during the daytime. They will only open the stomata during the night time. So this kind of stomata, the stomata which opened at the night and closed during the day, this kind of stomata, we generally call it as what stomata, friends? Sunken stomata. There's no need to get confused with that point. Now, all the students pay attention. Now, again, quickly recall my words. Let's talk about this stomata open, which means night time. So, what is the function of stomata? Everyone of you commonly know that exchange of the gases. Now, you look into this. Again, the carbon enter inside. So, who is the carbon acceptor in the C3 pathway? RUBP. Who is the carbon acceptor in the C4 pathway? PEP. Who is the carbon acceptor in the CAM pathway? Undoubtedly, it is also PEP. There's no doubt about it. Now, you only look into this. So, PEP is how many carbon? We are already studied that 3 carbon. So, PEP, which is a 3 carbon, accept 1 carbon from the atmosphere, which is 4 carbon, and giving rise to what, friends? Oxaloacetate. How many carbon it is? Which is the 4 carbon. Now the question is very, very clear. Who is converting or who is involved in the production of oxaloacetic acid? The enzyme which we generally call it as the PEP carboxylase enzyme. There's no need to confuse about it. Now, and very important point, you all people has to remember, when this entire reaction is happening only during the night time. And friends, that oxaloacetic acid, which is a 4 carbon containing compound, it can be converted into malate. Malate is how many carbon? Again, it is also 4 carbon by an enzyme called as malic dehydrogenase. Then that malate converted into malic acid and stored in the vacuole, because vacuole is the storage organelle. This is all happening where? Only during the night time. And what happens to the daytime? Now, because our ultimate goal is that we have to fix the carbon dioxide into glucose, which means that the plant has to prepare its food. I have told to all of you multiple times, whether it is a daytime, whether it is a night time, doesn't matter. The plant has to prepare the food every single time. So when the stomata got closed, we all know very well the carbon never comes inside. So what the plant will do? Whatever that is already stored, the plant start using that only. So now you can see that whatever the malic acid that is stored in the vacuole during the night time, that malic acid come out and it is converted in the form of malate. Now malate is how many carbon? We know very well. There's a 4 carbon. Again, the malic enzyme break down the malate into 3 carbon plus 1 carbon. Now, who will take this particular 1 carbon? Undoubtedly, it will undergo the Calvin cycle, because as we discussed that, Calvin cycle is the most common cycle. Now, another very important, what about the 3 carbon containing compound? Most of the time, the pyruvate will be remain as it is. Otherwise, friends, 80% of the cases, it will be remain as it is. Otherwise, it will be converted into the starch, which is a storage polysaccharide. Now, I am asking you a very simple question to all of you. My question is, when the pepper carboxylase enzyme will be in the inactive mode, will it be in the inactive during the daytime or will it be inactive during the night time? My question is very simple. When the pepper carboxylase enzyme will be completely inactive, I am asking. Inactive. Undoubtedly, there's no point of thinking it will be inactive during the daytime, because friends, there's no point of there's no function of the pepper carboxylase enzyme only during the daytime. Then there will be no requirement. So all the students look into the picture. There's no need to get confused. So this is called as a diurnal regulation of the pepper carboxylase enzyme. All the students, please look into this picture very correctly. This is the pepper carboxylase enzyme, which is in the inactive form during which time? During the daytime. And this is the pepper carboxylase which is active during the night time. And very important point for the exam that they don't ask. What they are going to ask in the exam, during the active form, will it be in the phosphorylated or non-phosphorylated? That is the question for the exam. Every student look into the picture. Pepper carboxylase enzyme will be super active during the night time. So friends, it will be phosphorylated. And we all studied multiple times. Serine, threonine, tyrosine. These three are the three major amino acids that undergo the process of phosphorylation. We all know very well. Now you only look into the picture. Serine is the amino acid that undergo the process of the phosphorylation, which time? During the night time. By that, the pep carboxylase will be super active. But during the daytime, please mute your videos compulsory. I'm telling you last and final time, if I find something, unprofessional in the class, I will immediately stop the class. Okay. Always maintain the class decorum. You are not 10th standard students to say multiple times. Always maintain professionalism in the class. Okay. Now, listen, every one of you. So if you look into the pep carboxylase during the night time, it will be completely phosphorylated. So then only it will be active. And if you look into the pep carboxylase during the daytime, it will be completely inactive. There's no doubt about it. Now, the question for the exam is very important one. Who is going to phosphorylate the pep carboxylase? That is important for the exam. Remember, every one of you, pep carboxylase kinase enzyme. Please don't get confused with the name. Pep carboxylase kinase is the name of the enzyme which add the phosphate group to the another enzyme, that is the pep carboxylase. Then pep carboxylase, how it get inactivated by see, because it once it get removed by the phosphate group, obviously it get inactive. How it will get inactivated? Someone has to remove the phosphate group. So who is that enzyme? Pep carboxylase phosphatase is the enzyme. So the question is like this: pep carboxylase phosphatase will be involved in the inactivation of the pep carboxylase enzyme. Pep carboxylase kinase involved in the activation of the pep carboxylase enzyme. These kind of statements makes you super confused. I want you to think properly and answer. Okay, every one of you quickly answer the question very correctly. So 4 mark 1. And the question asked is incorrect. Do not turn on your audio or video. Slowly think it properly. Okay, all of you. Okay, do not turn on the videos, please. Okay, now which one of the following statement regarding the pep carboxylase is incorrect options? All the students listen. During the day, they are asking a question. Understanding the statement is important for the exam. They are asking which is incorrect. During the day, C4 pep carboxylase is inactive, whereas CAM pep carboxylase is active. Now all the students has to tell me why it is incorrect. Everyone tell me why it is incorrect. Every one of you tell me why it is incorrect. Because friends, everyone, during the daytime, CAM pep carboxylase enzyme should be inactive, is it? So that is the reason the entire option will be the wrong option. Okay, we got that answer. Now, pep carboxylase is inactivated by dephosphorylation. That is what? True statement. Yes. Pep carboxylase kinase phosphorylates the pep carboxylase. Very correct. Statement 3 also absolutely correct. Now all the students look into the fourth statement. It is common statement. The synthesis of a pep carboxylase kinase is modulated by the circadian rhythms in the CAM leaves. Shafia, that's a common statement here. What is the meaning of a circadian rhythm here? Circadian rhythm is explaining about the day and night. Now you try to understand the given statement here. What the statement says that the synthesis of a pep carboxylase kinase enzyme is controlled by the day and night. Is it or not? Of course, yes. Hence, the fourth statement is also a true statement. The correct answer will be the number 1. There's no need to get confused with that. That's good. So that is about what, friends? CAM pathway. I hope every one of you understand it very correctly. Now, let me quickly tell you a little bit about the compensation point. And I want you to remember that every one of you focus. Compensation point is a point. There will be a no loss or no gain. Every one of you remember there will be a no loss or no gain of a carbon. Now, in other words, listen. See, the compensation point basically we can represent with a symbol called as I minus. I minus is, every one of you listen. I minus nothing but the carbon dioxide concentration at which there is a no, listen everyone, where there is a no net uptake or loss of carbon dioxide. That is what exactly we can call compensation point. I can tell you in a simple words, friends, the rate of carbon fixation equal to the rate of carbon loss. That is what we simply call it as the compensation point. So the compensation point will be the 0. And every one of you quickly answer my question. If you take a C4 plant, if you take a C3 plant, every student answer. Among the C4 and C3 plant, which plant will be more effective in fixing the carbon? Which will be more effective in fixing the carbon? Okay. Now, can I tell you one simple statement to all the students? Can I tell you one statement? Even though the carbon dioxide concentration is low, can I say a statement? Even though the carbon dioxide concentration is low, the C4 plants are highly effective in fixing the carbon when compared to the C3 plant. So if the C3 plant wants to fix the carbon should be in the highest concentration. So what is the conclusion here? The conclusion is that C4 plants have a lower compensation point than the C3 plants, because even though level of carbon dioxide is very low, the C4 plants are designed in such a way that by the help of Cron's anatomy, they can fix the carbon very easily. Now you can ask me a question, sir, what is the reason that you can say that C4 plants are always having the lower compensation point than the C3 plant? I can tell you two reasons. One is the Cron's anatomy, and another one is they are C4 plants are suppressing the photorespiration. That is the reason C4 plants are more effective compared to the C3 plants in terms of the carbon fixation. Don't get confused with that. So that is what the dark reactions, which we have started in the yesterday class and we finished it. Now, and before completing the photosynthesis, we are left with a little more topic, a little more important one, that is photoprotective mechanism. I already explained you the photoprotective mechanism, so I don't focus on multiple times now. I'll focus only on one time. I want you to understand it very correctly. Okay, then before that, a very important one we have to focus on: physical separation of the two photosystem. The statement itself is a tricky statement. What is the statement is actually talking? See, every student focus. This is the okay, you already seen this diagram, right? You look into this diagram. We have seen this diagram. This is the thylakoid membrane. Now you can see this. This is the photosystem 1 and this is the photosystem 2. Now my question is very clear. Why electrons are not directly flowing from the P680 directly to the P700. Why? The electrons are always flowing via several electron acceptor molecules. What is the reason? So remember my words. If the electron is directly flow from the PS2 to the PS1, it is impossible. You can't generate the energy. You can't produce the NADPH. You can't produce the ATP. So now the point is what? It is very important. P680 and P700 has to be physically separated. When their location is physically separated, friends, then only it is possible. Then only it is possible that the electron flow occur via series of electron acceptors. All the students, please look into the diagram. There's no point of getting confused here. This is the diagram showing this is the stroma. What is stroma? Fluid filled cavity. You can see that. What was this? Thylakoid membrane. And what is this? This is called as the lumen. You got the point very correctly. Now all the students focus. This is the LHC. I strongly believe every one of you remember what does exactly the LHC means. LHC means it made up of antenna pigments. It made up of reaction center. I hope you believe, you remember. This is the PS2. Good. And this is the cytochrome B6F. This is the PS1. And this will be the ATP synthase enzyme. All the students look into the picture very correctly. Look into this. Look into this. Now, if you look into the diagram very, very clearly, the light green color one, what is that? It is the PS2. Now, you only look into the picture where exactly the PS2 is located. All of you pay attention. PS2 is located predominantly in the staked regions of a thylakoid membrane. So what is the meaning of a staked region? If you look into the diagram very carefully, friends, you look into this. The PS2 is tightly attached, tightly arranged between all these thylakoids. Can you see any kind of a photosystem too that is freely arranged? No, it is arranged very tightly between the thylakoids. That is what we can call the staked regions of the thylakoid membrane, which is tightly attached. Now, if you look into the PS1, if you look into the ATP synthase enzyme, you only look into this. This is the PS1 and this is the ATP synthase. Can you see the PS1 and ATP synthase are tightly attached? No, they are extremely loosely attached. So what I can say that PS1 and ATP synthase are found in unstaked regions. What is the meaning of a unstaked regions? Non-oppressed thylakoids. What is the meaning of a non-oppressed? Where the thylakoids are not at all associated tightly with each other. Simple point. Now look into the cytochrome B6F. You can see this, right? Blue color one. You tell me where the cytochrome B6F is present. It is present on the loosely arranged thylakoids also and tightly arranged thylakoids also. You know what is the statement I can tell? They are evenly distributed. There's no point of getting confused. PS2 arranged tightly. PS1 ATP synthase arranged loosely. Cytochrome B6F evenly distributed. That's all. It's a simple type of a arrangement. Okay. Every one of you answered the question very correctly. I think something you have asked, no, right, Mauli? I think, is this the question? Yeah. Every one of you answered the question, the same question, Mauli. Yeah. Everyone. Following statements are related to the organization of a 4 major protein complexes of the thylakoid membrane. I want every one of you to answer it. Okay. All of you. Others are unable to answer. Why? Okay. All right. So you look into this, look into this very correctly. Photosystem 2 is located predominantly in the staked regions of the thylakoid membrane. That is right. Photosystem 1 is found in the unstaked, which is loosely arranged. Cytochrome B6F is confined. Correct, na? Okay, got it. Cytochrome B6F is complex is confined to stroma. That is wrong, right? Because it is evenly distributed, right? ATP synthase is where it is located. It is located in the unstaked regions protruding into the stoma. That is also right. Only the option C will be the wrong. Hence, the correct answer will be the option 2. Right? That's perfect. So I have covered entire photosynthesis without leaving anything except photoprotective mechanism. See, the people who are preparing for APPSC and SET exam, you people no need to read that. Okay, you people can leave it. CSIR people compulsory read, mandatorily read photoprotective mechanism, which I have already explained 2 to 3 times to all of you last time. Okay, compulsory read that. Okay, because it takes a little more time to solve other topic also. If I find the time at the end of the class, I'll teach you. Okay, now we will focus on the next a very important subtopic. That is what we can call as nitrogen metabolism. So again, I don't say that this time we can get the question or not. I cannot say that. But what I'm observing is in the 2023 paper we got in the morning shift also, afternoon shift also. Just now a TS set exam is over. In that set also it asked, and last year set also that is asked. Before that also it is asked. So in the CSIR also it is asked more than 7 to 8 times. But in the only CSIR, it is very special. They may ask, they may not ask. But I can say that out of 100%, 95% of the chances are there that we can expect the question in the exam. Okay. Just now I told you, right, Akshita, you're not listening my words. Right. Okay. Now listen. Next one, nitrogen metabolism. Whenever you are reading the nitrogen metabolism, listen what exactly you have to focus on. First point, I'm telling you, first point, focus on the processes. Second, focus mostly on the bacteria. And very, very, very important, where we are getting a very difficult type of questions in the exam, that is only from the enzymes. So far, whatever the question that I have seen little tricky and difficulty in the nitrogen metabolism, that is only from the enzymes. So I am requesting all the students to give a highest importance to the enzymes. So do not disturb me until half an hour. After 8 or 8:05, I will give the time. You can ask all your doubts. Okay, focus. It's not too difficult topic. It's interesting. I'll do little faster. Okay, nitrogen metabolism or nitrogen cycle, that every one of us know very well. No living organism on this planet Earth cannot survive without nitrogen. It is impossible. The reason is that nitrogen is exclusively required for amino acid, protein. It is required for DNA, required for the RNA, required for enzymes. Multiple organic molecules are exclusively depends on the nitrogen. But what is the extreme disadvantage of this particular nitrogen? The nitrogen which is present in the atmosphere, we all know very well. We use it to say that the nitrogen present in the atmosphere is 78%. Huge percent of nitrogen is present. But unfortunately, whatever the nitrogen that is present in the atmosphere, it is chemically inert. More specifically, it is N triple bond N. We, animals, humans, plants, no animal, no human, no plant do not have any kind of a enzyme that break this triple bond and they can make use of the nitrogen directly. So now what is the advantage and what is the disadvantage? The major disadvantage is that no animal, no human, no plant cannot break it. But we can't survive without nitrogen. So someone has to convert this this inert nitrogen into most simple forms of the nitrogen. What was that simple forms? We generally say, right, NO2, NO3, NH3, NH4 plus. These are all said to be the most simple forms of the nitrogen. So we can't convert the atmospheric nitrogen into the most simple forms. So someone has to convert it. Who is going to do that? Undoubtedly, bacteria will do it. Bacteria do that, give it to the plant. From the plant, animal will get. Upon death of plant and animal, again soil will get. From the soil, again we will send it to the atmosphere. So now what's the point you all people has to understand? Why did we use the word nitrogen cycle? Because there is a cyclic movement of the nitrogen. Every one of you pay attention. All the students focus on the diagram. There is a cyclic movement of the nitrogen from one tropic level to the another tropic level throughout the ecosystem. That is the reason we call it as nitrogen cycle. There's no point of getting confused at all. Now, all the students follow me very correctly. There are many steps are there. First step is called as nitrogen fixation. Without understanding this slide, you can't understand the concept at all. First, what is the meaning of a nitrogen fixation? I want to convert the atmospheric nitrogen into the most simple form. What is the simple form? Either NH3 or NH4. That process, we call it as nitrogen fixation. So there is no point of confusion. Nitrogen fixation is the first step. But you tell me, finally, a bacteria converted the nitrogen to NH3 or NH4. But what is the point? Do you really think we can use the NH3, NH4 directly? No. They are also little toxic forms only, friends. So again, some more bacteria, what they will do, they convert NH3 or NH4 to further simple forms, such as NO2 and NO3. And this second step, what do we call it as? Nitrification. Now the question is crystal clear. Why we converted N2 to NH3, NH4? Why we converted NH3 or NH4 into NO2 and NO3? What is the reason? We have to assimilate. What is the meaning of a assimilation? Assimilation means what? Incorporating into the body, utilizing in the body, making use of it. So finally, after nitrification, assimilation. Who will assimilate first? Of course, animals cannot assimilate first. Plants will assimilate. Assimilate means plant is using, utilizing it. When the plants start utilizing it, plants will produce their proteins, their amino acids, their nucleic acid, everything. Now we, higher tropic level animals, from the plant, we will take that particular proteins, amino acids. Correct, na? Upon eating the plants, we animals get. So animals and plants are getting the nitrogen in the process of assimilation. What is the first step? Fixation, nitrification. Third step, assimilation. Now you think properly. Plant died, animal died. So what is the meaning of a death here? What happens after the death? Everyone know that after the death, dead organic matter can be converted into inorganic. Now the question is very, very clear. What is the meaning of a dead organic matter? Dead means what? Proteins, amino acids, nucleic acid, they are all dead organic matter. What is the meaning of inorganic? We are converting all into carbon, hydrogen, oxygen, nitrogen. Correct, na? So friends, the point is simple. Once plants and animals die, the decomposers, what they are going to do? They are going to break down the dead organic matter and they will use the nitrogen. Because friends, of course, bacteria also compulsory require the nitrogen. Without nitrogen, bacteria also can't survive. So bacteria also use the nitrogen. Now what it will do with the extra nitrogen? That extra nitrogen, bacteria will fix in the soil in the form of what? Ammonium. I'm using the word bacteria always fix excess nitrogen. I'm using the word excess. Excess nitrogen in the soil. That is called as ammonification. Now you only try to understand who got benefited now. Plant got benefited, animal got benefited, is it? And microorganism also got benefited. But you tell me, from where we all people has taken the nitrogen? We have taken the nitrogen from atmosphere, which means that we have to maintain the balance compulsory. So we are sending the nitrates back to the atmosphere. That is called as denitrification. So what you can understand by this slide, the cyclic movement of the nitrogen from one trophic level to the another trophic level throughout the ecosystem. That's what we exactly call it as a nitrogen cycle. Never get confused with that. Now, focus. The question won't go beyond whatever I am teaching you. Make sure that you listen me properly. So, first steps, nitrogen fixation. They will ask the question from the equation also. What is nitrogen fixation? N2 can be converted into NH3 or NH4 plus. What is nitrification? Conversion of NH3 or NH4 into NO2 and NO3. That is called nitrification. What is called assimilation? Conversion of NO2, NO3 into amino acids, proteins, other nucleic acid. So assimilation means utilizing, converting into utilizable form. What is called denitrification? Conversion of NO3 back to the atmospheric nitrogen. And I have already told to all the students last time, so far, I'm using the word so far. So far, friends, we have identified only 5 bacteria. That's all. So far, we have identified only 5 bacteria which can convert NH3 directly to N2. I want all the students has to understand me. Actually, in the process of denitrification, what's happening? I am converting NO3 back to what? N2. Correct? Now, back to N2. But we found out 5 bacteria so far, they are having the enzymatic system which will convert NH3 to N2. Never get confused with that particular point. Now all the students look into this beautiful and simple diagram. First, what happened? Atmospheric nitrogen. Atmospheric nitrogen in the form of a rain. It is reaching to where? Soil. Who will fix in the soil? Bacteria. Correct na? Then bacteria only convert them into more simple forms, NO2 and NO3. And again, what we are doing? We are bringing this NO3 back to the atmosphere. Don't get confused. That's why it is always keep on cyclizing. If the nitrogen is not cyclizing, all the trophic levels never get the nitrogen. So it is very important to undergo the cyclization. Never get confused. Now, what we have to read in the lesson? We have to read all the 5 steps. You know, what I used to observe is, this topic is very easy and rigid topic. 90% of the students will make the wrong answer, will give the wrong answer, because they'll get confused with the given statements in the exam. So that's why you focus on the statements correctly. Among the 5 steps, the most important step is nitrogen fixation, assimilation. These 2 steps are very important steps where we can expect a more number of questions. Let's start one by one. The nitrogen fixation. Okay, before that, answer every one of you this TS set question. The exam which was happened few days back. Very simple question. My voice is clear. If anyone, am I clear for everyone? Refresh, keep refreshing, man. Okay, I believe every one of you will answer the question. Okay, answer. This is the question they have asked in the set exam few days back. Every one of you answer. Slowly. You're giving so fast answers. I don't want that fast answers. Okay. Everyone, others also, everyone give the answers. Oh, already given. Great, great, great. You can see it, right? Atmospheric nitrogen, lightning, rain wash, nitrification. Correct, na? So first, always the nitrification, assimilation, ammonification, denitrification. Such a simple question they have asked. I don't know how many of people got answered it correctly. Okay, now let's focus. Let's focus. Okay, now let's focus on the first and the biggest step. I am requesting every one of you not to interrupt me up to 20 to 25 minutes. Let me complete the complete step, because at least one or two statements in a given question comes from the fixation. So I want all the students has to spend, pay proper attention. Focus. Atmospheric nitrogen has to be converted into NH3 or NH4. That is our ultimate goal of a nitrogen fixation. Now, every student commonly know that we can fix the nitrogen industrially also. We all studied during the +2 by a Haber's process. Everyone know that. We also fix the atmospheric nitrogen fixation. Just now you see in the question by natural lightning. That is just 5 to 8% only. But friends, what we people has to focus on, we no need to focus on atmospheric nitrogen fixation. We no need to focus on industrial fixation. We have to focus on bacterial nitrogen fixation. So if the nitrogen fixation wants to done, we require a versatile enzyme. What was that versatile enzyme? Nitrogenase enzyme. I don't want you to focus on the any chemical processes, not required at all. We exclusively focus on biological. Now, friends, now the question is clear, no? Why plant cannot fix the nitrogen? Why animal cannot fix the nitrogen? Why humans cannot fix the nitrogen? Because no one having the nitrogenase enzyme. That's the reason only. So now who's gonna fix the nitrogen? Undoubtedly, bacteria is gonna fix. Now again, this is a tough challenge. You all people has to remember the name of the bacteria compulsory. There are two type of bacteria that are involved in the nitrogen fixation. One is called free living, another one is called symbiotic. All the students commonly know that symbiotic bacteria are those type of bacteria always maintain a relationship with the other organism for its growth and development. Now, free living means, so it is in case of a symbiotic bacteria, it is in the relation with another one, and both are helping each other, and they are involved in the nitrogen fixation. But if you look into the free living, it won't dependent on anyone. It will grow on its own, it will develop on its own, and it is having the ability to fix the nitrogen. The point is very clear. Both free living bacteria and symbiotic bacteria, both bacteria having what enzyme? Both bacteria having the nitrogenase enzyme. Never get confused with the statement. Now, all right, look into this. Again, after the class, I will send you the questions which compulsory, contains the questions from the bacteria names. I believe everyone will answer those questions accurately. So that's why focus on every single example, because we will get the questions from example. You know why I'm stressing like that? Last time we got the questions from enzymes. Before we got the questions from genes. Before that we got the questions from process. Again, we got the questions from enzyme. Now there is a possibility you can expect the questions from bacteria. That's why you should not neglect that. Here, nothing to explain, you have to by heart. There are symbiotic and free living. Again, no need to confuse and no need to remember everything. Focus on some more important ones, especially leguminaceae plants. Remember, Parasaphonia, groundnut. These are all the plants which give the or which maintain the association with the all rhizobium species. Azorhizobium, Brodyrhizobium, Photorhizobium, Cynorhizobium, all these. And remember the examples correctly. Casuarina, only remember the casuarina, enough. Alder. Alder is a rubber tree, everyone commonly know that. Casuarina and alder. Frankia is the name of the bacteria. Gunnera, nostac. Azolla, anabina, sugarcane, acetobacter. I want you to remember 5 friends. These 5 bacteria are very crucial. Now, what previously they asked in the exam? So remember, these are all symbiotic fixing bacteria. And what are the free living? Cyanobacteria. Blue green algae, every student commonly remember the cyanobacteria. Hence, no need to focus on that. Whichever you already know, don't focus on that. Focus on aerobic, facultative, anaerobic. In the question paper, they asked Anabina, Acetobacter, Bezeranschia, Darkshia. These 4 examples they have asked in the previous exam. I want you to focus on all. What are aerobic? Everyone commonly know that the bacteria compulsory require the oxygen for their growth. What about the facultative? Basically, they do not require the oxygen. Without oxygen also they can survive. There's no doubt about it. But whenever there is a presence of oxygen, their growth will become more. Their development will become more. In the presence of oxygen, the survival rate is more compared to the absence of oxygen. But in the absence of oxygen, they will grow. And what about the anaerobic? Everyone know that if there is a oxygen, they will die. So they are strictly independent, sorry, they are strictly not dependent on the oxygen, which is the anaerobic bacteria. Again, in the tomorrow class also, I'll ask you the first question from this table. Make sure that you remember, okay, compulsory. Now focus on this. Now we are in the first step, that is we are converting N2 to NH3 or NH4. Requesting all of you pay super attention from this particular slide. Understand every single statement that I'll explain you. Now listen to me carefully. What is the major enzyme? Nitrogenase is the major enzyme. This enzyme having one disadvantage. What is the disadvantage of this enzyme? So where this enzyme is present? This enzyme is present inside the free living bacteria also, inside the symbiotic bacteria also. This enzyme is present. Now what's the point? This enzyme, if there is a high level of oxygen, this enzyme automatically get inactivated. Remember my words. This enzyme sensitive towards the oxygen. Presence of oxygen will inactivate the subunit of the nitrogenase enzyme. By that, the nitrogenase enzyme will get inactivated. Don't get confused. So, all the students remember, whether it is a free living bacteria, whether it is a symbiotic bacteria, whenever they are using this enzyme, they have to create an environment where there should not be oxygen. In simple words, the free living bacteria and symbiotic bacteria has to maintain anaerobic conditions compulsory. That's the crucial point. If they are not maintaining the anaerobic condition, the enzyme never work. Now let's see. Let's see what are the special type of mechanisms shown by the bacteria. Let's talk about the cyanobacteria. What is that cyanobacteria? Free living bacteria. Now the free living bacteria, which is the cyanobacteria, please turn off your videos. Don't disturb me in the middle. Okay. Now, I don't know why what is happening here. All right. Okay. So now listen. Cyanobacteria. What is the specialty of a cyanobacteria? You can see this, right? Cyanobacteria developed a specific structure. What we generally call it as, friends, we generally call it as the heterocyst. Now, my question to all the students is very simple. The heterocyst will be found on the bacterial cell wall every time. My question to all of you is, the heterocyst will be found in the bacterial cell wall every time? No. All the students remember my words. This particular heterosis can be developed by the bacteria only certain times whenever it is required. So friends, it's a very important statement. Additional cell walls with glycolipids. You know what is the specialty of this heterocyst? It contains lot and lot of glycolipids. And what is the specialty of the glycolipids? They are not allowing the oxygen to enter inside. Such an important statement it is. It is not at all allowing the oxygen to enter inside. Another point, very crucial point, they do not contain photosystem 2. We all people studied multiple times. Wherever the photosystem 2 is present, what is going to release? Oxygen is going to release, is it? So, but this heterosis maintain the conditions in such a way that there will be a no PS2 will be present. Hence, there will be a no release of the oxygen. Now, my question is crystal clear to all the students. So cyanobacteria do not have the PS2. We got that point. Now you can ask me, sir, then how does the cyanobacteria can synthesize the ATP? Because PS2 is very important for the ATP production. That's a very good question. So instead of the PS2, they depends on PS1. Very important one. And very important point all the students has to remember, the cyanobacteria for the food, I am talking about the food, they exclusively depends on the neighboring cells. Please don't confuse with this particular point. This is just one mechanism maintained by the cyanobacteria. What is the ultimate goal? They make sure that the nitrogenase enzyme present inside, it will be active all the time. All the time means whenever they want to fix the nitrogen. Now, this is one mechanism. What is this mechanism called as? Anaerobic condition. What is that condition? Development of heterocyst. Then the next one. Now, this is the difficult one, right? There is a bacteria called as aerobic nitrogen fixing bacteria. Name itself confusing us. The bacteria, so example, this is a azotobacter, this is a Gliotiaceae. If these two bacteria want to grow, want to develop, compulsory oxygen is required. Without oxygen, they can't survive. Now, inside the azotobacter and Gliotiaceae, there is a presence of a nitrogenase enzyme. There is no doubt about it. Now, the question is very clear. If they want to fix the nitrogen, how? How it will happen? Because if they want to fix the nitrogen, there should not be oxygen. If there is no oxygen, they will die. Then what is the condition? How they can avoid this condition? Remember, I am using a word that they maintain reduced oxygen condition. I am not at all using the word oxygen is completely absent. They maintain reduced oxygen condition. An example, suppose example, if the oxygen is required 50%, they will reduce the oxygen percentage to up to 10%. That's all. Only for particular period of time. During that time, oxygen level will be dramatically fall down. Then nitrogenase enzyme will be super active. It fixes the nitrogen very fastly. Then again, after that, it will start using the nitrogen normally. So it will maintain reduced oxygen condition only during a particular period of time. What is the specialty of a Gliotiaceae? It evolve oxygen photosynthetically during the day. We know very well, during the day only light reactions are happen. So oxygen will be releasing out. So during the night time, they fix the nitrogen. So somehow the Gliotiaceae azotobacter make sure that nitrogenase enzyme will be super active. How they are making it active? By reducing the level of oxygen in their body only during the time of nitrogen fixation. That's all. Then what is the meaning of a facultative organism? All the students remember, they generally fix the nitrogen under anaerobic. There is no problem only because facultative can survive easily in the absence of oxygen. That is the advantage. Anaerobic, of course, if the oxygen is present, they only won't survive. So for anaerobic bacteria also, there is no problem in fixing the nitrogen. So friends, in order to make you confused, they will ask a very simple question that no one can answer. Which of the, you will get, by looking at the option only, you will confuse. Which of the following bacteria are efficient in nitrogen fixation? Now you tell me what answer you people will give me. So I am just showing you these three conditions. Which of the following bacteria can show the efficient nitrogen fixation? Yes, everyone. There's no need to confuse, right? First anaerobic, then followed by the facultative. They can fix the nitrogen in a very efficient way. Never get confused with that point. Okay, all the students focus. Try to understand my statements very correctly and keep on answering my questions very accurately. Please focus. What is our goal? Our goal is that activating the nitrogenase enzyme. Correct, na? Where the nitrogenase enzyme is present? It is present in the bacteria. Now, who require nitrogen? First of all, plant require nitrogen. So, if the nitrogenase enzyme, which is present in the bacteria, if it wants to be active and it has to be useful for the plant, first, bacteria and plant has to maintain the relationship. Without relationship, how the nitrogenase enzyme fix the nitrogen? Now listen, establishing symbiotic relationship. Again, the previous statement that they asked in the exam, the symbiosis between the legumes and rhizobia is not obligatory. Such a good statement it is. They will give the statement that the relationship between the bacteria and plant is always obligatory, which is a wrong statement. Bacteria and plant never maintain the relationship all the time. They will only maintain the relationship whenever there is a requirement. If there is no requirement, they never maintain the relationship. Now the condition is plant require nitrogen. So plant require nitrogen, so it has to maintain the relationship with bacteria. So that's why you see the point what we have written. Symbionts. Symbionts means both plant also and bacteria also. Seek out elaborate exchange of the signals. When they are exchanging the signals, only under such an important point, nitrogen limited conditions. When the nitrogen is limited, then only they start maintaining that particular relationship. Now the question is clear, very clear. Why they want to maintain the relation? What is the ultimate significance of maintaining the relationship? Ultimate significance of maintaining the relationship is root nodule formation. That is the ultimate goal. Now, almost all our questions comes from genes and enzymes. Now focus on the genes. If the root nodule want to be formed, there is a what is meant by the nodule? Nodule nothing but group of cells. If the root nodule want to be formed, there is a requirement of two type of genes: noduling genes, nod genes. Nod genes widely called as nod factors. Correct, na? Nod factors. Now remember all the students. Noduling genes and nod genes both are exclusively required for the root nodule formation. But where they present is entirely different. Noduling genes specifically present in the plant. Nod genes specifically present in the bacteria. Never ever get confused with that point. Now, the interesting point is that if the nod genes are the nod factors, where they are present within the bacteria, there is a condition. What is the condition? If the nod genes or nod factors which are present in the bacteria, if they want to get activate, there should be a activation of another gene. We call it as nod D gene. What is the meaning? What is the speciality of a nod D gene? It is a constitutively expressed gene within the bacteria. Now, if you ask me, sir, what is the meaning of a constitutively expressed? All the students imagine, just think it. In case of our body, protein synthesis will be happening 24/7, 365 days until we live. The protein synthesis is very common, which means that the ribosomal RNA will be active all the time. The ribosomes are also active all the time. So their genes are constitutively expressed in our body. It is mandatory. Similarly, NOD-D gene within the bacteria, it should be constitutively expressed. Now you can ask me, sir, what is the relationship between the NOD-D gene and NOD factors? The point is that NOD-D gene is the one which will activate the NOD genes. So remember, all the students remember, I can simply tell to all of you, NOD-D is the regulator. We can get the same statement in the exam. NOD-D gene is a regulatory gene which regulate all other NOD genes. So how nicely it is activating. Look into this. Look into this. We will discuss in the upcoming slide, but listen. NOD-D genes, see, there are many NOD genes are there. You can see, right? So listen everyone. How all these NOD genes are getting activated. See this. If all these NOD genes wants to be activated, all the students look into this. There will be a box. Very important statement. We called NOD box. What is this NOD box? It is a promoter, friends. It's a promoter. It is a promoter. How does this promoter is getting activated? This promoter is getting activated whenever the NOD-D gene will come and attaches. Now you see this, right? Whenever the NOD-D gene will come and attaches, NOD box will be activated. When the NOD box will be activated, every single NOD gene is going to get activated. Very simple point it is. No need to get confused with that. Now, this comes a very important statement. So first, NOD-D gene is going to activate the NOD genes or NOD factors. What are the type of NOD genes or what are the type of NOD factors? I don't encourage all the students to remember the structures. You don't get it. But I want you to remember the structural properties. First of all, how many NOD genes are there or how many NOD factors are there? All the students remember there are two type of a NOD genes are there. One is called as common NOD genes. All the students remember. One is called common NOD genes. Another one is called host specified NOD genes. Correct na? Now, what are the common NOD genes example? A, B, C. That's all. What are the host specified NOD genes? P, Q, H, F, E, L. Now you can ask me a question. Sir, actually the common NOD genes and host specific NOD genes both are present in the bacteria only. Both are required for the nitrogen fixation only. Then what's the difference? The difference is that, as the name itself suggesting, right, common NOD genes. My friends, with the common NOD genes, nitrogen fixation is not 100%. Nitrogen fixation is not 100%. But whereas hosts, I can say that they are the generalized genes. If the nitrogen fixation wants to be more efficient, we always use the host specific NOD genes only. Never get confused with this particular point. Now, all the students look into this picture. What are the common NOD genes? A, B, and C. And the statement for the exam, which we got in the exam. NOD factors or NOD genes, whether it is a B, C, P, Q, H, E, F, L. All the students remember my words. They all are lipochitin oligosaccharide signal molecules. Such an important statement it is. They are lipochitin oligosaccharide molecules. And this is the first statement they have given, and this is the second statement they have given in the exam, as we discussed. they are having a beta 1,4 NAG and NAM linkase. But in the exam, they have given a statement that they have a chitin alpha 1,4 linkase, which is a wrong statement. Always remember, we all know, right? Chitin is a polysaccharide, friends, which is always having a beta. Never ever get confused with that point. Now, all the students remember. Is there any necessity to remember the structure? I don't want. But I want you to read which carbon is attached to the which carbon. That is important for the exam. As a four marker question, as one of the statement, we can expect that. So it's mandatory to know that. Right. Now, what about the PQHFEL? What I told to all of you, they are extremely required for a specific host. Never get confused with that one. Okay. Now, all the students focus on this diagram. So just focus on 15 minutes very clearly. I'll explain the mechanism, then I'll solve the MCQs, then we will take a little bit break. Okay, focus. Now our ultimate goal is symbiotic relationship. What is the ultimate goal of a symbiotic relationship? Maintenance of the nodule formation. How is this possible? Every students look into this beautiful picture. All the students look into this. This is called root hair. All of you listen. So this is the bacteria, okay? Whichever I am drawing in the blue color, that is the bacteria. Within the bacteria body, what is there? What is there within the bacteria body? NOD D gene is there. And within the bacteria body only, NOD genes are also there. Correct, na? Now, every one of you use your brain. So what happened? The plant starts releasing certain chemoattractants. Again, there is a statement. Don't think that the plant is going to release the chemoattractants every single time. No, the plant is going to release the chemoattractants only whenever they require. So now they want to attract the bacteria compulsory. So they released the flavonoids. Now those flavonoids will go and attract NOD gene. The moment it attract the NOD gene got activated. It's a very powerful one. Now the NOD gene, look into that. It will come and attaches to the promoter. Where? Promoter. The moment it attaches to the promoter, every single NOD gene will get activated. Now you only look into that. Now that NOD factors, they will initiate the root nodule formation. Now you can see that. Now I will explain you step by step how exactly the root nodule got formed. So this 15 minutes are very crucial. Pay attention. It's super easy. Listen, all of you. This is the root hair, right? The root hair of the plant starts releasing certain chemoattractants. Chemoattractants means which attracts the bacteria, which we can call the flavonoids, isoflavonoids, and betaines. They're all chemoattractants. Now they attracted which one? Bacteria. All the students look into the diagram. Now this is the bacteria, right? Small ones, rhizobia. You only look into the picture. Now the rhizobia will come and attaches to the root hair. Now the question is very clear. How is this possible? How does bacteria will come and attaches to the root hair? That is the question. My friends, this is the root hair. Exactly, root hair look like this. On the root hair, there are many receptors are there. All the students listen to me correctly. On the root hair, many receptors are there. You know what do we call those receptors? Lectin receptors. Now, those bacteria will exactly come and attaches to that lectin receptors. So friends, no need to confuse. I will repeat it. First, bacteria will release the root hair will release the chemoattractants. Chemoattractants attract the bacteria. Bacteria will come and attaches to the lectin receptor. Lectin means sugar. Sugar receptors present on the root hair. This mechanism is called as RICA adhesion. What is the mechanism? The attachment of the bacteria to the root hair. That is called RICA adhesion. Now look into this beautiful picture. The moment bacteria got attached, what's happening? You only look into that. Bacteria immediately cause curling of the root hair. Now the question is very clear. Why they have to curl the root hair? Without curling, they can't. Without curling, they can't enter inside very easily. So they want to make a passage. They want to make a way in order to enter inside. That's why they will cause curling of the root hair. The moment they cause curling of the root hair, all the students focus. Now you can see it, right? Bacteria can easily move inside. So friends, whenever the bacteria is moving inside, you know what do we call it as? Infection thread. What do we call? Infection thread. Now bacteria is moving inside, right? Now all the students understand the important point. When the bacteria is traveling inside, you know what happens? Bacteria lose its cell wall. Suppose friends, this is the infection thread. Okay, bacteria is moving like this inside. During the movement, bacteria lose its cell wall completely. Upon losing its cell wall, it is surrounded by a plant-derived membrane, which we called as rhizoids or bacterioids. I will explain you step by step. All the students focus. Focus everyone. First, plant root releases chemoattractants. Then they will activate NOD-D gene. Then the NOD-D gene activate further all the NOD genes. Then bacteria come and attaches to the lectin receptor by a mechanism called as RICA adhesion. And they cause curling of the root hair. Why they cause the curling of the root hair? Because they want to enter inside. That's an important point here. Whenever they keep on moving inside, they keep on infecting more plant cell. When they keep on infecting more plant cell, they lose their cell walls. When they lose their cell wall, they can be surrounded by a plant-derived membrane that is called as bacterioids. That bacterioids only fix the atmospheric nitrogen. Now you can see this, right? Every one of you look into this. This is the nodule. Our ultimate goal of the nitrogen fixation is what? To fix this particular, to synthesize this root nodules. What is the speciality of a root nodules? What is the significance of this? Every student look into this. Gas, such an important statement. You know, we can expect the similar kind of statements in the exam. Gas permeability is regulated in root nodules. Enough oxygen to carry our respiration. See, such an important statement it is, but low enough to prevent the deactivation of nitrogenase. So you have to understand the speciality of a nodule. It is allowing the oxygen only in a required amounts. It never ever allow the oxygen in excessive amounts, because if the oxygen will allow in excessive amounts, what happens? Nitrogen fixation never occur. So how much amount of the nitrogen is, oxygen has to be passed? Only it will allow that much amount of the oxygen only. That is the significance of this root nodule. Now you know very well the root nodule contain pink colored pigment. You know, right? Hemoglobin is actually a red color. But every student focus. Here the hemoglobin is in what color? Pink color. Now you can ask me, sir, why did it get the pink color? Because friends, it is present in the infected cells. It is not present in the healthy cells. Is it? Since it is present in the healthy infected cells, the leg hemoglobin will get a pink red color, but mostly it is in a light pinkish in color only. Now what is the ultimate goal of this leg hemoglobin? What is the ultimate goal? Look into this. Leg hemoglobin having the highest affinity for oxygen. So whenever the leg hemoglobin associated with the oxygen, our nitrogenase enzyme will be protected. Then the nitrogenase enzyme never be inactivated. It will be keep on fixing the nitrogen whenever it is required. So in one single word, I can tell to all the students why the root nodule formation occurs between the plant and bacteria is just to synthesize this leg hemoglobin. This leg hemoglobin is very powerful in such a way that it is having the highest affinity for oxygen. That is the reason the root nodule formation will occur. Anyway, now ultimately root nodule formation occur, our nitrogenase enzyme will be free from oxygen. Now listen, this is the most crucial step. Nitrogenase enzyme is ready now. We are ready to fix the nitrogen. Now the question is crystal clear. Who is ready now? Nitrogenase enzyme is ready. Now, if the nitrogenase enzyme wants to be activated, it is also required a regulatory gene. What is the name of the regulatory gene that can fix the nitrogenase enzyme? That is called NIF gene. Never forget. You remember, I told you that NOD-D gene is the regulatory gene for every single NOD gene present in the bacteria. Similarly, NIF gene act like a regulatory gene for the nitrogenase enzyme. Now, ultimately, our nitrogenase enzyme is ready. Okay. Till now, till this slide, whatever we discussed, you may get the question, you may not get the question. But trust me, from now onwards, every point is important. We will expect the question. You only see how twisted statements they will raise it. Now, every student know that nitrogenase enzyme complex is ready now. What is the function of nitrogenase enzyme complex now? It has to fix the N2 to NH3. This is only our ultimate goal of a nitrogen fixation. Now, it is mandatory that every student has to remember the composition of every single enzyme that we come across. Focus. Now, see friends, I'll just show you. So this is the nitrogenase enzyme. Okay. Every point is important, friends. It contains two metalloprotein complexes. This word also important. What are the two metalloprotein complexes? One is called as denitrogenase reductase. Another one is called dinitrogenase. Simply remember, one is called reductase. Another one is called dinitrogenase. Instead of that, I will also tell you to remember in a simple way. Nitrogenase is a two metalloprotein complex which contain iron sulfur redox center and molybdenum iron redox center. In a previous set exams, I think it is a Madhya Pradesh set, they have asked a question. They have asked a question that which of the following cofactors required for the activation of the nitrogenase enzyme? So it requires iron, it requires sulfur, it requires molybdenum. Right? Remember, three are required, but the major one is the molybdenum followed by the iron. Don't forget. So listen, the nitrogenase enzyme complex is separated into iron sulfur protein, molybdenum iron protein. Now, is it really necessary to remember the subunits? It is mandatory to remember the subunits. I just want to show you, okay, we should always learn from our previous year questions. Now, this is the question which was happened, paper happened few days back. You see this, in this question, what they asked, see the question. They have given 4 enzymes present in the electron transport chain of mitochondria. They did not ask anything about inhibitors. They just asked about the subunits. That's all. That is the direct question they have given in the exam. Similarly, in our exam also, they used to ask the questions very commonly from the subunits. So nitrogenase enzyme having 4 subunit. That is very important, friends. Now, all the students focus on the diagram. From the starting of the lesson, I am repeatedly saying to all of you, nitrogenase enzyme will be inactivated when there is a presence of oxygen. That is the statement I'm saying repeatedly. Why? That is the question. The answer is that nitrogenase enzyme having 2 subunits. This is the first subunit. This is the second subunit. The first subunit named as dinitrogenase reductase. Look into this. It is extremely sensitive to oxygen. So friends, this subunit, if there is a presence of oxygen, it will get inactivated automatically. See an example, because it is an enzyme, friends. Suppose this is the enzyme. If I increase the temperature up to 55 degrees centigrade, what happens? The active site get damaged. Similarly, when more oxygen, the active site of the enzyme get damaged. That is the reason we are repeatedly saying that nitrogenase enzyme is inactive when there is a presence of oxygen. Now I am going to explain you the final step. Pay attention for 5 minutes. What's our goal? Our goal is conversion of N2 to NH3. But the point is very clear. You can't fix the nitrogen directly into NH3. It is impossible. You can't fix the nitrogen into NH3 just like that in a single step. How many steps it will take? All the students has to focus on the diagram very accurately. If you understand the diagram, all concept is very clear. It takes 3 steps. Step 1, step 2, step 3. Okay. Now you listen to me. N2, it is getting converted to NH double bond NH. That also we will expect in the exam. Then first step. Then NH double bond NH converted to NH2 NH2. Then final step, NH2 NH2 converted into NH3. You got that. You got that. Now there are 3 steps are there. I'm giving you one example to all of you. Every single step, there is a requirement of 1 pair of electron. Every single step. Remembering electrons is very important for the calculation part. Okay. There will be a requirement of how many pair of electron? 3 pair. Every step, 1 pair. Now the question is super clear. Everyone focus on the diagram very accurately. This is nitrogenase enzyme. I am just explaining you, friends. Okay. This is the enzyme, right? This is how an enzyme look like. What is this? Active site. To the active site, who will come and attaches? Substrate. Who is the substrate here? Nitrogen only substrate, no. Because we have to convert the nitrogen into NH3. Now the question is very clear. If the N2 want to attach to the active site, we require 1 more pair of electron. So my friends, my point is very clear to all of you. Nitrogen, if it wants to attach to the enzyme, it requires 1 pair of electron. And once again, nitrogen, once it attaches, if the nitrogen wants to convert it into NH3, there 3 pair of electrons are required. So total how many pairs of electrons are required in order to convert N2 to NH3? Total 4 pairs, which means 4 into 2, 8 electrons are required. 4 into 2, 8 electrons are required. Now, that is also not the question. From where these electrons are coming from? That is the crucial point here. That's the crucial point. Now, every one of you focus on the diagram. Try to understand the diagram very accurately. Focus, be patient. Look into that. See this? This is the nitrogenase enzyme. This is the subunit 1. This is the subunit 2. Correct na? Who is the electron donor? Very important point. We can get the question directly in the exam. Ferredoxin is the electron donor. The question was asked also. Ferredoxin is the electron donor for nitrogenase enzyme. So ferredoxin donate 1 pair of electron. Every student commonly know that. Whenever the ferredoxin donate 1 pair of electron, it will undergo the process of oxidation. Yes or no? Losing electron is called as oxidation. Now you tell me, who is going to accept this 1 pair of electron? Iron sulfur protein is there, right? That is only accepting that 1 pair of electron. Now that iron sulfur protein is getting reduced. Now tell me, that iron sulfur protein, do you really think it will keep the electron with it? No, it has to send to the molybdenum iron. Then again, it will also get oxidized because it is donating the electron. Now who will get reduced? Molybdenum iron will get reduced. Now tell me, molybdenum iron, do you think it will keep it? No, it is sending the electron to N2. Then again, it will also get oxidized back. Then N2 getting converted into NH double bond NH. Just one step is over. Again, second pair of electron is coming to iron sulfur protein, coming to molybdenum iron. Finally, it come to where? NH double bond NH. It is converted to NH2 NH2. Again, the third pair of electron is coming. Iron sulfur protein, molybdenum iron, finally coming to NH2 NH2, and it is getting converted into two molecules of NH3. So now three pair of electrons are there, correct na? And one pair, why we used? One pair is attachment of the N2 to the enzyme. There one pair. Total how many pair? Four pairs. Now all the students has to follow me correctly. What I have written the statement here, I have written a statement that the dinitrogenase reductase enzyme, every student follow me, it contains two ATP binding sites. What's the meaning of that line? What is the meaning of the line two ATP binding sites? Which means whenever an electron passes, it will convert ATP molecule to the ADP. In simple words, it will cause the hydrolysis of the ATP. Simple point. Now, one electron, every student has to follow me, friends. If one electron, just one electron I am talking, one electron. If one electron passes through the iron sulfur protein, it hydrolyzes two ATP molecules to the 2 ADP. But we all discussed just now how many electrons we are using. Total 8 electrons we are using. So one electron hydrolyzes 2 ATP, which means that 8 into 2, 16 ATP molecules are getting hydrolyzed into 16 molecules of ADP. That is the reason in a given statement, they always give a statement. Nitrogen fixation is a energy consuming process. This is the first statement they use it to give in the exam. Of course, true statement, because we are spending 16 molecules of ATP. That's why it is a very, what we can say that energy consuming process. Now you see the overall equation, N2, 8 electron. To maintain the charge balance, we will take 8 protons. We are spending 16 ATP, then N2 reduced to NH3, 16 ATP reduced to the 16 ADP. This is what a simple nitrogen fixation. Okay. Every one of you, please respond to me. Is it clear for everyone? I hope I am not going fast. Even though I go fast, we have to, cope up. Of course. All the chemoattractants belongs to the secondary metabolites. There's no doubt about it. Correct, na? Now, I strongly believe every one of you will give me the right answers. Okay. Now, every one of you focus on the given question and answer the question correctly. I need an accurate answer. Okay. Answer, everyone. Be patient. Please spend 30 seconds. Stop giving me spontaneous answer. I don't want your speed answers. Just read it. They're asking true statements. Slowly. What about others? Others. Come on, be active. Bindu, what would be the answer? Chaitanya. Okay, good. Okay, all of you focus on the given options. Nitrogenase enzyme is composed of two components, dinitrogenase and dinitrogenase reductase. That is true statement. Molybdenum iron protein is a component, is a homodimer, no? And iron protein is the component of dinitrogenase, no, right? Iron protein is the component of dinitrogenase reductase. So option C is also wrong. Molybdenum iron protein contains the active site metal cluster where N2 binds. Right statements. Iron protein delivers electrons to molybdenum iron in a reaction coupled to the hydrolysis of ATP, which is catalyzed by the magnesium. That is also true. Hence, the correct answer is number 4. Very good. Okay. Take your time. I want you to give me the correct answer. See, knowing the concept is not important, friends. Remembering the concept is important. Okay. Always focus on remembering. Everyone. Okay. Take time. Take time. Okay. Okay. I believe that you are all people are reading the question at least for 30 seconds. Is it? Please read the question for at least 30 seconds. Okay. Don't neglect it. Because of your excitement, you will choose the wrong answers. Tell me, all the students, why the option C is wrong option. I want every one of you to tell me why the option C is the wrong option. Why? So because both are dependent. So first, iron sulfur is donating the electron to the molybdenum iron. Is it? Both are working cooperatively, we can say that. Correct, na? Hence, option C is the wrong answer. Hence, the correct answer is A, B, and D. All right. Okay. Okay. Oh my God. Time is already 8:37. Okay. Okay. Time is 8:37 right now. Oh, 8:38. Okay. Take the break for a half an hour. 8:40 means okay. Exactly. I will start the class at 9:10 p.m. Sorry, 9:10. Exactly 9:10. I'll start. We will finish that topic. Okay. Sharp 9:10. I'll share the another link to all of you. Okay. All right. Thank you all. Thank you.